Hi, this is Nick Cap, and this is the second part of the lecture for Chapter 1, kind of an introduction to microbiology. Some of the main topics that we want to cover in there would be basically looking at the history of microbiology. What I wanted to emphasize for you in this particular class is that even though we're covering the health sciences and microbiology for the health sciences, ultimately, when you're looking at this, you are a scientist. You are kind of looking at why. And sometimes in the health sciences, what you're looking at is just we need to get a patient better. But ultimately, to help everyone get better, we need to know why. Why does this work? And that really prevails itself in us looking at our drugs, our vaccines, and everything else. Why do they work? And again, in this time of COVID, you may be like, why is it taking so long to do a vaccine? Is number one, we want to make sure that it's safe, and number two, we want to make sure that it works. So we need to do something that we call the scientific method. The scientific method has a number of different steps. The first step is basically to make an observation. Does this work? Or you look at something that's happening. And again, scientists publish a lot of their kind of observations of what they're doing. Then you make a hypothesis. If I take a piece of a virus, a piece of the SARS virus, and purify it, can I inject it into somebody and it will make a vaccine? Part of this is on other observations, but that's a hypothesis. You then need to test the hypothesis and really truly see that it works. Not just like, oh, I'm pretty sure it works, or give it to a friend and tell me that it works. No, we need to really test that it works. And a lot of times we do like a double-blind study where we don't know what's going on, and that's why we need to do that. And then you need to draw your conclusions on that. You need to take the data, look it in, analyze it, look at the conclusions, get people from outside to help you analyze it. And then, again, on repeat, we need to repeat this to make sure that we know that it works. And so, again, during this time of COVID, do the masks that you have work? And again, we look at in the very beginning, and I did a podcast on this. People thought, oh, we don't really need to use masks. Masks don't really work all that yet. Yet, really, with the social distancing and wearing a mask, it reduces it just enough so that it works. And one of the problems that we may have is that, again, we didn't really do the scientific method on the mask, and we poo-pooed the mask, and now a lot of people don't want to wear the mask, and they're like, you scientists told us a while ago that the masks didn't work. And again, it wasn't really the scientists, we weren't really doing the methods. And so, again, practice your scientific method. The requirement for the scientific methods are basically that you're looking at a single variable. You can't take a whole bunch of different things. And what I mean by variables is you're only looking at kind of one thing. And so basically you can't say something as complex as cancer is caused by one thing or another when you're just looking at kind of one variable. And I'm not doing that well. So basically in single variables is a lot of times we don't, on things like looking at the environment and does a particular procedure work? What we tend to look at is just, are we just looking at the bacteria, or are we looking at a number of different things in there? We need to do experimental controls. That is, who is getting the example that I use a lot for that is my elephant repellent. I'm wearing my elephant repellent right now, and it's working fantastic. There's no elephants around me. But trouble is, again, there's no elephants here, so we need to have a positive control. You need to do something where a result is going to happen because it's there. Again, we'll talk about a little bit, class, we may think about a little bit, is that how can this be used to discover things when we look at this? And again, the cause of HIV causing AIDS, the cause of SARS causing COVID, etc., are all things that we look at. It's important to know the knowledge of microorganisms. Our knowledge of microorganisms have availed us to do things like prevent food spoilage, to prevent diseases from occurring, although this one kind of slipped through, but we haven't had a pandemic in over 100 years, so that was pretty good. Our knowledge of microorganisms led to aseptic techniques that prevented contamination in medical and microbiology laboratories. Again, the CDC has universal precautions, and we'll talk about this a little bit when we come into the lab, is basically use gloves, gowns, masks, and goggles, these barriers that are outside. You minimize the risk of needle sticks. Again, you don't really touch the needles. We have safety containers that we put sharps inside. the disinfection procedures that have been scientific proven to work, a preventative treatment after exposure, so that again you do to get exposed to something, you take antibiotics or something like that in order to do that. Overall, you need to reduce the risks. And again, that's one of the things when you shelter in place, we're not going out, we're not increasing the risk of catching the particular disease. And lastly, we kind of treat all the patients the same. Don't have a patient come in and say, "Oh, you probably have SARS. I'm not going to touch you." You know what? We need to go and treat all of them exactly the same. Somebody with a pinprick could have a disease that we can catch, and we should really do all that. And so, in looking at it, we've been gotten kind of lazy the last few years and not doing our preventative precautions. It shouldn't be something that we put up, this barrier that we put up on just certain patients. We need to have that. Again, I'm a lot worried of hepatitis rather than HIV or SARS. So one of the things that we need to look at in this class is spontaneous generation. And so the hypothesis that living organisms arise from non-living matter is what we call spontaneous generation. And according to spontaneous generation, this vital force forms life. And there is a vital force of life, but it's not causing life to form from nothing. And really, a lot of this was us not doing the scientific method in the very beginning, in the 1800s. An alternative hypothesis is that living organisms arise from pre-existing life, and that's what we call biogenesis. And Pasteur was one of the people who kind of did a simple, elegant experiment in order to show that biogenesis doesn't work. And so Pasteur, what he did is he put some broth inside this a flask. He would take this long neck flask and pull the neck over. He'd boil the material, kill everything in there. And if you leave these flasks, kind of nothing would happen to it. You break off this long neck, and it would grow almost instantaneously. And again, people were talking about vital forces, and when you had this open long neck here, it would be exposed, and the vital forces could go through there. And so, again, Pasteur won an award for doing this. Where's microbiology currently being practiced? And this is one of the things that we will kind of talk about in class is, again, a nurse, a doctor, a respiratory therapist, a surg tech. You're practicing microbiology all the time, but microbiologist is practiced by people in, and actually we talked about it in the veterinary field for the job that you apply to, and educational settings, of sanitizing when you do that, as well as all the microbiologists in the Bay Area that are employed by biotech companies that are discovering some of the drugs and new foods that we're eating. A timeline of microbiology, we're going to go over this in class, and on Wednesday you're going to make a timeline. The golden age for microbiology, in a time that I wish I was alive, but it's just as exciting now, is from the roughly the 1815s to 1910s. And this is the beginning of Pasteur's work. Discoveries included the relationships between microorganisms and disease. We were discovering what causes disease. For this first time, we were looking at immunity. We were looking at antimicrobial drug. Pasteur didn't invent fermentation, but he really did kind of help it. Pasteur showed that microorganisms are responsible for fermentation, for the production of beer. He was really concerned with the production of wine. Fermentation is conversion of sugar to alcohol to make beer and wine. That's one example. Microbial growth is also responsible for spoilage of food, and Pasteur looked at this as well. Bacteria that use alcohol and produce acetic acid spoil wine by turning it into vinegar. So if you don't want vinegar and wine turns into vinegar, that's kind of a bad thing. This is a great picture of Pasteur. If you go visit us on campus, we have a nice little statue of Pasteur. He is one of the giants, and he's one of my favorite scientists. Pasteur demonstrated that the spoilage bacteria that could be killed by heat, that was not enough to evaporate an alcohol in wine. This application of high heat for a short time is what we call kind of pasteurization. And so what we're looking at is a lot of times is these pathogens get killed a lot faster than some of the other ones. And so when you pasteurize something, you reduce the number of microorganisms that are there. You're not necessarily sterilizing it. For the germ theory of disease, 1835, well over 100 years ago, Bassi showed that silkworm disease was caused by a fungus. In 1865, Pasteur believed that another silkworm disease caused by a protozoan, and again, the production of silk in textiles is one of the big motivators of there. In the 1840s, Ignaz Semmelweis advocated hand washing to prevent transmission of pleural fever from one OB patient to another in pregnancy. So basically what would happen is doctors would go in and practice cadavers, and they would go and deliver babies, and they wouldn't wash their hands in between. Things that we think are really disgusting now, people were really hesitant of doing. In the 1860s, Joseph Lister used a chemical disinfectant to prevent surgical wound infections after looking at Pasteur's work showing that microbes are in the air can spoil food and cause animal diseases. 1876, Robert Koch provided proof that a bacterium causes anthrax and provided the experimental steps Koch's postulates used to prove it. And again, at this time in 1876, people were dying of anthrax all the time. We had a lot of horses and stuff like that around there, and this particular disease was present at the time. Chemotherapy is the idea of using kind of chemicals to kill one organism and not the other organisms. And again, it's treatment of chemicals is what we call chemotherapy. Chemotherapeutic agents used to treat infectious diseases can be synthetic drugs or antibiotics. Antibiotics are chemicals produced by bacteria and fungi that inhibit or kill other microbes. Quinine from the tree bark was long used to treat malaria. And so, again, we would take these natural preparations and isolate that individual drug, identify that individual drug that worked at it. In 1910, Paul Ehrlich developed a synthetic arsenic drug, Salvarian, to treat syphilis. And in the 1930s, other particular drugs were identified. A lot of people have heard about Alexander Fleming's discovery of penicillin. It wasn't necessarily a fluke because he was actually looking for these kind of things. He observed that the penicillium fungus made an antibiotic penicillin and it killed S. aureus. And so this fungus is growing in here, and when he streaked out the bacteria would have this zone of inhibition. This bacteria should grow there. In the 1940s, penicillin was tested clinically and was mass-produced. The big impetus for that was the World War II, and that people really needed this material for that. Modern developments in microbiology. Bacteriology is the study of bacteria. Mycology is the study of fungi. Parasitology is the study of protozoa and parasitic worms. We tend to not have that as much. There's not a big call for parasitologists in the United States. Recent advances in genomics and the study of an organism's gene have provided new tools for classifying microorganisms. And again, one of the reasons why we were able to identify the SARS bacteria that causes COVID-19 is by using these techniques, and we will talk about this a little bit later. The Nobel Prize, you guys have all heard about, are these kind of awards for doing good work in science, although there's a bias in that, definitely. 1901, the diphtheria antitoxin was discovered. And again, this some of what you may hear about is serum therapy for COVID-19 that was used at about this time. Ross looked at malaria transmissions. Koch was the TB bacterium. Minchnikov discovered bacteriophages. Fleming penicillin in 1945. Wakesman streptomycin in 1952. So antibiotics were big for a while. Dolberg, Hershey, and Loria were looking at viral replication in 1969. Tanagawa is looking at antibody genetics, and Prusner is looking at kind of prions. And you will be looking at a timeline of this biology. And what I encourage you is to kind of not just look at necessarily these. Go out and find the scientist who could be you, who looks like you, and kind of see what they do. And if you can get some, find somebody who you think kind of inspires you, and I think that would be good. In all of science, everyone is important. And again, we tend to take people like Ross and Behring and Koch and put them on a high pedestal. But there's a number of human endeavors that go into kind of picking who those people are. And for every Michnikov that's in there, there's many people that are in the backgrounds that had a hand in kind of helping with them. In microbiology, we're going to be looking at some units, the micrometer, the nanometer, angstrom. We're going to be talking about this in lab this week. Basic parts of the microscope include the eyepiece, base, condenser, eyes, diaphragm, objective lens, and again, we'll talk about that during lab today. Not lab today, it's during lab this week. We will need to look at magnification calculation. Magnification is the objective power versus ocular power, and we'll talk about that. You need to know that microscopes are parafocal, they're paracentric, and you need to be able to know how to measure something in the microscope. Again, in order to see something, you need to study it a lot. And this is an example of some bacteria in here. And next week what we're going to do is we're going to have you come into the laboratory and we're going to verify that you know how to use the microscope. And so this Wednesday we're going to go into that a little bit more, and next week we'll go into a little bit more. All right, this has been Nick Cap. It's been very fun talking to you about the history of microbiology. I know I didn't go into it a lot. We're going to have you guys do an exercise where you kind of discover some of this history yourself. I know it's hard to memorize some of these names and who the people are, but go ahead and try and do it, and you just might see some inspiration from doing it. Catch you later.